Thank you so much, everyone, for coming today. This is my pleasure to have Dr. Sala for this DocSig webinar series, who will talk about this automated, free-to-use analysis software pipeline that should facilitate our analysis of PET scan data. It's very important, particularly because we published recently with Michele Farisco a paper showing that one of the most challenging parts of the guidelines that has been published is really the implementation of multimodal assessments. And so the PET scan, as you know, is a big help in trying to diagnose and differentiate level of consciousness. And so that's a big deal that a group or someone thought about developing that pipeline for analyzing these data. So Dr. Sala, thank you very much. I will introduce you and then you can start, okay? So Dr. Sala is a postdoctoral researcher at the Coma Science Group, led by Aurore Thibault and Olivia Gossery. Dr. Sala's work aimed to improve the understanding of the pathophysiology of disorder of consciousness. Dr. Sala uses multi-level molecular neuroimaging assessment to investigate patterns of hypometabolism and brain disconnectivity in patients with disorder of consciousness based on PET combined multivariate method of analysis. It seems very complicated, but I know that Dr. Sala will try to make it as friendly as possible for us to understand how we can implement that as clinicians, but also to understand what are the evidence around using the PET as a very useful tool for differentiating disorder of consciousness. Dr. Sala?
Okay. I will share my screen. So first of all, thanks a lot for the invitation. Actually, today, it will not be that hard. So now today I will focus really on the easier massive univariate analysis. So it's not molecular connectivity. So I hope it will be understandable. So you can see my screen, right? It's all right. Yeah, okay, perfect. So I will so I know PET, positron emission tomography, is kind of an exotic technique. So many people don't know much about PET. Some people don't even know that it's still used. So I would like to give a very short introduction about positron emission tomography in general. Then I will move towards using positron emission tomography in disorders of consciousness. And then I will finally present the pipeline with a tutorial. So I will show you how to actually use it, what files to check for interpreting the results. So for positron emission tomography, just very briefly, so we're talking about a neuroimaging molecular techniques that is based on radioligands. What I really care about for these slides is just I want to say that PET measures can measure many different things. So we have different radioligands, and depending on what we inject, we can take a look at several different aspects of brain biology. So we can, of course, measure glucose metabolism, brain perfusion, and we can go very deep into understanding neurotransmission, both at postsynaptic and presynaptic level. And we also have a lot of targets for brain pathology and neuroinflammation. So it's a very diverse neuroimaging technique. It's a bit different, I think, from many other neuroimaging techniques or electrophysiological techniques, because the temporal scale of PET is rather slow. So when we do a PET imaging, typically we acquire images for around 10 to 15 minutes, so 720 seconds. And we don't have like in fMRI, the image is not like, it's not a video. So there is no temporal component. What we have is just an average of whatever happened in the brain over these 10 to 15 seconds. So we are much slower than fMRI, and we are much, much, much slower than EEG. And we are, of course, so what we are looking at is, the sum of many thousands of neural events happening in the brain during the full scan. At a spatial level, we actually are not doing so bad. So the spatial resolution right now of a PET scanner is around 4.3, 4.5 cubic millimeters. That is not so different from fMRI, which is around 3. And what it means? Well, what it means is that when we look at a box cell in a PET image, we're looking at around, well, the sum of whatever is happening in around 2 million neurons. For comparison, we're talking about 800,000 neurons in fMRI per voxels, and 100 million neurons if we look, you know, at say the spatial resolution of EEG. So it's much more integrated. So PET is much more, let's say, precise compared to electrophysiological techniques. I think maybe it's not very well known, but PET is also the neuroimaging technique that allows us to look at brain function, and that really covers the full brain. So in fMRI, in theory, can also do that, but typically based on the typical acquisition protocols that are used, that doesn't happen because there is a space left between the slices that we acquire, so we don't really cover the full brain. And also very important to stress is that PET has a good spatial resolution at cortical level, but it's also very good at measuring radioactivity at the level, subcortical level, at the level of cerebellum, and at the level of brainstem, allowing us to really have a, let's say, accurate picture of whatever is going on in all different regions of the brain. So we don't have issues, for example, for regions that are at the air tissue interfaces, which happens, for example, with fMRI. And I think it's very important to show you how a PET image look like, because I think many people, if you say PET, they will tend to think about these kind of images. This is an image from the 90s, which is like, okay, yeah, I see some blobs of activity. This is actually not how images look like today. So right now, and this is not any special scanner, it's a pretty, standard good scanner. So the images that we have and the spatial resolution that we have is more in this order of things. So this is just as an introduction to PET, as I'm not sure if a lot of people are familiar with it. So what's the application of PET in the OC? In the OC, so I showed you there is many different targets that we can target with PET, different biological processes that we can target with PET. But with disorders of consciousness, I would say that, I guess we're really at the start of things. There is just a few tracers that have been used in association with PET in disorders of consciousness, most commonly FDG for the assessment of glucose metabolism. So what is FDG? FDG, it's an analog of glucose that allows us, it's a tracer, so it allows us to track the activity of glucose in the brain without, of course, altering the activity. So it's a glucose labeled with an 18-fluorine isotope. And why do we care about it? How glucose is related to neural activity? Well, when we inject our FDG, this FDG gets metabolized by exokinase and converted into, what the analog of glucose 6-phosphate, which represents the first step of glycolysis, which can be oxidative metabolism in neurons or aerobic glycolysis in astrocytes. And the very important thing is that, so we use glucose to produce energy, of course, to produce ATP, but the production, the usage of glucose is linked by a specific biochemical pathway to glutamatergic neuronal activity. So when there is some excitatory activity going on, the usage of glucose is stimulated in the astrocytes. And it has been shown in an animal model using stimulation of brain activity via a laser that let's say dynamics, the changes in the calcium concentration in the neurons, so that is considered a more direct index of neural activity, synaptic activity. So synaptic activity is strongly related to the use of ATP, so of energy consumption with a bit slower neural temporal dynamics, but still we have a strong coupling between glucose metabolism and neural activation, with more than 75% of glucose metabolism being associated to tasks related to neural communication and neural activity. So why using FDG-PET, let's say, in patients with disorders of consciousness, compared, for example, to fMRI? So there is some advantages of using PET. First of all, we don't have to be worried about metallic implants. So PET doesn't use any magnetic fields, so it's 100% compatible with respirators or whatever metallic tools are being used on the patients. Right now, the radiation exposure, so we rely on radioactive tracers, right? But the radiation exposure is like 3 to 4 times lower than, I would say, 20 years ago. So right now, here in Liège, we inject around just 100 megabecquerel, which exposes the subject to 2.5 millisievert, which is equivalent to the radiation that every one of us gets in 1 year, just based on the natural radioactivity in the environment. The assessment, the scan duration is short. Right now, we do around 10 minutes for a scan, but with a new scanner, we will probably be able to take it down to even less, maybe 8, 7, 6 minutes. And sedating the patient to avoid movements is not an issue. This is because when we inject, so we do the injection, then we have to wait 30 minutes before putting the patient in the scanner, because the current protocols use simplified measurement and acquisitions procedures. So we inject the patient. We don't need to do any kind of sedation at the time. We can keep the patient under control, make sure he's awake, and under control conditions, and only after 30 minutes we put him in the scanner. And after 30 minutes, the uptake of glucose of the tracer is has reached a plateau, so we can apply sedation without affecting the results of the exams in a significant way. So what kind of measures we have available for, has been used, let's say, in patients with disorders of consciousness? Well, it's 2 types of measures. The first measure refers to the global absolute metabolism, as you can see here. So this is quantified in different ways. For example, using standardized uptake value, SUV, which is the simplest measure we can use to quantify the absolute consumption of metabolism globally in the brain. And why we care about the measure? Well, it has been proved that glucose metabolism, absolute glucose metabolism changes in different conditions. Even in sleep, we have a decrease of up to 20-25% of our global consumption in glucose metabolism. And in disorders of consciousness, the decrease observed is pretty strong, in the order of 50 to 70%. So let's say it's quite a huge effect that kind of, let's say, tends to give quite clear results. And it has been proved that with good accuracy, one can distinguish between unresponsive patients and minimally conscious patients if we consider a decrease of 58% as a cutoff between the 2 clinical groups. Another measure that we can use, I think it's actually, this has been used much more frequently, refers actually to relative hypometabolism, as you can see here. So relative decreases in glucose metabolism and relative preservation of glucose metabolism. So here we're not interested really at the global decrease overall, but we want to look at in each individual regions, at each individual box cells. So box cells are just the pixels of our images, 3D pixels. We want to look and localize where we have the strongest decreases in metabolism, and we have relatively preserved glucose metabolism. So of course, the full brain of this patient is going down. So overall, we have a decrease of 50 to 70%. But here we can look at the relative differences compared to a reference control group. And we can say in these regions, we have relative hypometabolism, so the strongest decreases in metabolism. And these other regions in red, we have the less affected regions, the relatively preserved regions. How can we do this? We can compute a metric called standardized uptake value ratio, SUVR, and typically we do the comparison because we need a comparison with the controls, of course, to say where the decreases are taking place. So we use a software called statistical parametric mapping, which is what you will see in the pipeline. And in particular, for the purpose of differential diagnosis, it has been shown that looking specifically at hypometabolism at the level of the internal and external awareness networks is particularly relevant. So we're speaking about the regions of the precuneus, of the cingulate, gyrus at a medial level, and at frontoparietal regions at a lateral level. And it's also been shown that subcortical regions like thalamus and the caudate nucleus are relevant for disorders of consciousness. So overall, we could say that we have a good match between, let's say, the level of consciousness and the level of glucose metabolism alterations in the different groups of patients, both at the level of absolute global metabolism. As you can see here, in blue, we have the lowest values observed in unresponsive patients, and let's say less strong decreases as the degree of consciousness improves. And same for relative hypometabolism. You can see here in unresponsive patients, we have the most extensive alterations in metabolism, with the most extensive decreases at the level of the internal and external awareness networks, while again, going up in minimally conscious patients, the extent of the hypometabolism is less strong. However, it has also been shown in subjects that appear as unresponsive that actually metabolism can be partially preserved, or let's say can be compromised not as much as one would expect in these patients that again present as unresponsive. And you can see here an example. And there was a paper published last year by Aurore Thibault, by our team, that showed that if we can categorize patients based on the degree of alterations of glucose metabolism. In particular, here they considered absolute glucose metabolism in the frontoparietal regions, and they were able to differentiate. So among the behaviorally unresponsive patients, they could differentiate between those that where they are defined as minimally conscious state star. So those that are behaviorally unresponsive, but their PET scan is not that as compromised as one would expect. And so we can differentiate between these patients and let's say the true unresponsive patients. And this is quite relevant because if we then look at a prognostic level, while all unresponsive patients had a negative outcome, either as resulting in death or as staying in an unresponsive wakefulness syndrome, minimally conscious state star patients actually had in 1/3 of the cases a positive outcome. So either progressive to minimally conscious state or even emerging in a minority of cases from minimally conscious state. So there is quite some evidence that PET, is useful in disorders of consciousness, and all this evidence and more evidence has resulted the most recent guidelines on diagnosis of disorders of consciousness now recommend the use of FDG-PET for the differential diagnosis of disorders of consciousness. And based on 5 studies and almost 400 observations, they show that there is a 95% sensitivity, 70% specificity in distinguishing between unresponsive and minimally conscious patients based on FDG-PET. Still, this evidence, again, is based on the type of metrics that I have shown you. So this is metrics that it's not something you just get. You do the PET scan, you get the images, and you just visually assess the raw images that you get. But these metrics require some data analysis, some processing, which is not necessarily available in all clinical centers. It requires quite huge, let's say, technical expertise. So our response to this issue is indeed this pipeline. So the idea of the pipeline, once again, is to try to reduce the amount of technical difficulties in generating the kind of metrics, again, the SPM maps, the maps of relative changes in metabolism, and in assessing the global SUV, so the global absolute changes in metabolism in clinical centers. So the pipeline solves, let's say, the technical difficulties, but still requires, let's say, sufficiently expert users to then interpret the results. So this is something, it's something that we cannot solve on the technical side. But still, I think it helps a lot already to remove these technical issues and also make things much more efficient than if you would do all these steps manually. No matter how expert you are, all these steps will take more than 30 minutes to do manually. With the pipeline, it will take more like 1 or 2 minutes to run. So how do these steps, how does the pipeline work? So the only thing that the user should do is really to select the images of the patient that as they come out from the PET scanner. Typically, it's DICOM images. So it's what you would get just exporting the images out of the scanner. Then the analysis is done following 2 different paths. So in one case, we want to get our SPM maps of relative hypometabolism and relatively preserved brain regions. In the second case, we want to get an estimate of the global SUV. So have an estimate of the global decrease in patients, again, in our patient, as compared to the control. So it's a pipeline, once again, for individual assessment, for individual diagnosis, for use in clinical settings. More into detail, just to give you a bit of an overview of all the steps that are involved here. So the pipeline, first of all, so we provide the image to the pipeline, and then everything is automatized. So first of all, we have the DICOM images are converted into a different format called NIfTY, and some useful information are extracted from our DICOM files about the patient and the PET scan. And these are all information that are important for estimating these SUV metric. Then all these parts pertains the spatial normalization. So the point here is that we have one patient with a very specific anatomy, and we want to compare this patient to a group of healthy controls. To do this, we need to put our patient and the healthy controls in the same space. So physically, we want to achieve as good as possible overlap between the brain of our patient and the brain of our controls that we want to use for comparison. This means performing a co-registration, and which just rigidly moves the image of the patient to match the average image of the controls, which is called here, it's a template. It is also provided with the pipeline, and it is a template that has been created specifically for this kind of analysis. So it's a DOC specific template. Then what happens is that our software, all of this is based on SPM, our software takes again our co-registered images and performs a spatial normalization. So essentially it matches using a series of affine and non-affine transformations our patient images to the control, this template image, making the 2 images perfectly comparable. Still, there is some variability that we tend to expect in patients with hugely deformed brains, as is often the case for DOC patients. So we apply a sort of smoothing. Essentially, it's kind of we kind of apply a filter, like an Instagram filter, to our image. To essentially, we decrease the level of detail of our image, but at the same time, we decrease also in inter-subject variability, so that our image is in the end more similar to our template and to all the control images that we want to use for comparison. Once this is done, so the patient image and the control images are in the same space, we finally run the statistical comparison with the controls. So we have to define a mask. So we ask SPM to create a mask of the regions of interest, that is our brain, for the analysis, and we run a statistical comparison. We then obtain our results, which means that we have maps that indicate, so this is a statistical comparison. These are T maps with T statistics that indicate where we have significant changes in the brain of our patients, either in terms of relative hypometabolism in blue or relatively preserved metabolism in red. Results are also breakdown based on different regions, macro areas, language, different resting state networks, and language regions specifically. And for each of these areas of interest, we also get an Excel file with an indication of the amount, the percentage, the proportion of voxels that are indeed hypometabolic or not in our patients. So this is the first step of the pipeline. So we get these relative maps and also these additional summaries to help the interpretation of the maps. Second, and this is the other pipeline, again, we have a very similar normalization procedures to, again, make things comparable between patients and controls, but we also compute the SUV images. So we take automatically some information from the DICOMs, our original images. Doesn't matter what scanner you use. So this information will be exactly the same for all Siemens, Philips, GE scanner. And we use them to compute an SUV image. So we perform the so-called absolute quantification. Then again, we compare our image. So this is an image that is an SUV image with the absolute quantification. It's in the same space as the controls. And we compare it to our controls using a mask of all gray matter to obtain a global value for SUV. And compared to the controls, we obtain the global, the value for the global decrease. So this is everything that the pipeline does. And again, it takes 1 to 2 minutes. If you do it by hand, no matter how skilled you are, it takes at least 30 minutes. So it's quite nice. Even if you know how to do this, it's nice to use it. I will run now a more practical tutorial to show you practically how to set this up and how to run it. So to set this up, you can go to, this link. So the pipeline is freely available on GitHub. And so you can just simply download it. Everything I will explain now actually is also present on the information on GitHub. So if you lose any steps, you can go back to GitHub. Everything is written there. For the setup, the setup is a bit complicated, but it's the most difficult thing on a technical level. Just once you need to set things up. What you need to do, you need first of all to install some required software. So you need to get MATLAB, you need to get SPM, and you need to install SPM. Installing SPM just means telling MATLAB where SPM is. And for this, I mean, there is a very nice videos of like 2 minutes on YouTube. You can just check it out. It's super, super easy to do. Then you need to get the code. Again, it's freely available on GitHub. So you just click here, click on local and download zip. And here you downloaded all the code. So here is everything you need. It's in the pipeline. This is the main script file that we will use when we run it. Actually, it's still not fully final because for a matter of say ethical consent, we couldn't upload the controls images on GitHub. So the control images that are used for comparison needs to be obtained from a different place, which is eBrains. So you have to go on eBrains and click on get data. You need actually to register and log in. But yeah, okay. So you just simply need to download the data. And the only thing you need to do is that in the folder you will download, there is a folder called data. You need to take this data folder, put it into the coffee break FDG-PET main folder. And now everything is there and really the setup is done and you can run your pipeline. So how it works. So I don't know if you're familiar with MATLAB. I will just briefly go over it. So here you have the command window where you type your commands. You will have to write just one thing here on the right. And on the left, you have the current folder. So what you need to do to run the pipeline is to go, here is very easy to navigate, to the folder that contains the code. So the main script that I showed before. So I select the first folder. You see here, this is the original folder plus the data folder that we got from eBrains. And this is the script that we want to run. So we just type coffee break FDG-PET, press enter. And this is the only thing you need to do. You need to select the directory containing the DICOM files of the patients. Here, how it works. Here on the right is just for visualization. Here on the left is for selecting things. You want to go here in the upper folder, so you click on the top 2 dots, and then you click on the folder that you want to go to. You then click on this point, and you have selected your folder. You click, and then it's going. So first of all, we are getting the information about, well, who was this person that take the PET scan. Here, of course, it's fake data to have it anonymized. Information about the PET scan, so how much we injected, how much the person weighs, which is very important. And here it's running. So now we have the script is importing the icons. And now the normalization phase, the spatial normalization phase has started. So now we have the co-registration, which is the part that takes the most time. So now it's running, will take a few more seconds. Okay, it's done. And you can see here the results. So here is the template that we use to co-register, and here is our image. You see that they are more or less aligned. Next is the spatial normalization. So now we are stretching our image so that each location really fits the template. And now already we are at the statistical comparison phase. We compare our patient with their controls group. Sorry, it's too fast. Then, okay, so we compare controls and patients, and we have our estimation of relative hypometabolism. And we have a generation of a series of renders that are saved automatically that you can use to look at, well, where the effects are. And here also in actual, at the actual level, we will see later, which is quite important for the quality check. Here is the second part of the pipeline. So now we are doing the absolute quantification with the computation of SUV. Again, there is a small part of spatial normalization once again, and that's done. So this took, I think, around 1 minute, 35 seconds, and all of this is done. So now, okay, I think this part is not the most difficult part. It's just you need really to click once, select your images, and then everything is taken care of by the script. But now the most important part, which is user dependent, starts. So we need to do quality check. We need to check if our results can be used as they are, or there's been some issues, for example, with normalization or with the mask that SPM generates to run the statistical analysis. So there is 3 things that we need to check in our results. First of all, we need to check the mask. So as I said, SPM generates a mask, which is supposed to be a brain mask that covers the full brain, based on which he runs a statistical analysis. So the mask is, we provide a mask, then we also ask SPM, well, take all brain regions that have a certain level of signal, and all the rest keep out because it's not gray matter. So for example, here you see in the mask, we don't have all the parts that are ventricles, cerebral spinal fluid. We also don't have skin, we don't have skull. So theoretically, this mask should cover gray matter and a bit of white matter. But you can see here, for example, actually we have a huge hole at the level of the left temporal lobe. Why this happens? Well, when a patient has a huge lesion or a huge decrease in glucose metabolism in a certain area, SPM might erroneously think that this area actually it's not gray matter. So this area is excluded from the analysis. What it means that when you look at these renders that I showed before about hypometabolism, this area will not pop up as hypometabolic, even if it is. So you need to be aware of this because you risk to underestimate the hypometabolism in the patient. So here you don't see a statistical effect just because this area was excluded from the analysis, not because there is no effect. Actually, this area has a very, very, very huge hypometabolism, so huge that it doesn't even look like we are analyzing gray matter. Second step, spatial normalization. So again, here it's very important to, as I said, we want to have that our image matches our template and matches our controls. So it can happen that this doesn't really work. It can happen especially in patients that are unresponsive, that have a huge atrophy with a huge enlargement of the ventricles. In this case, the caudate nucleus and thalamus tend to be really pushed on the borders, and the spatial normalization algorithms don't really work. So we can have a case, as you can see here, where this is our template, we are at the level of the caudate, and here we have just nothing. We don't have signal because the caudate is probably pushed on the side. So here we might have hypometabolism at caudate level in the results, but actually we cannot say anything about it because simply these two images in this specific area, they are not comparable because the normalization unfortunately failed at this specific level. So it's very important to look at the images and assess any possible issues with spatial normalizations that can occur, especially at a subcortical level, or in case, like in this case, of very huge lesions. Finally, the white matter. So what happens is that when we do our comparison, so in patients that have a very huge decrease in glucose metabolism at the level of gray matter, we might see some effects with relatively preserved glucose metabolism in the level of white matter. This is kind of an artificial effect that happens again when we have such a huge decrease in gray matter level, that the white matter might pop up as relatively preserved, because indeed, after we perform global mean scaling, which is something we do in these analyses, the white matter will just look as if it is even higher than in controls, but it's just a relative effect due to the full context, let's say. So why I say this, because if you look at the renders, so in the renders, the results are kind of pushed a little bit on the surface. So you might see sometimes that there is some relatively preserved metabolism in some regions, where you should open an actual render and check if that relatively preserved metabolism actually is not located in the white matter, which doesn't have any clinical significance. So you need to be careful about it. So how does it work? So the interpretation. So we check all these things, and then we can interpret our results. Again, here are two prototypical examples of an unresponsive case and minimally conscious state case. Again, in a responsive case, we expect very huge decreases in SUV, in absolute metabolism, less strong in minimally conscious state. We expect a huge, extensive hypometabolism encompassing most of the internal external awareness networks. That's not the case for MCS, where it should be less widespread. So how does it work? Here we are back again to MATLAB. So these are our results. So automatically the pipeline will change folder and bring you to the folder with the results. Here in DICOM, you have the original images. You have then the images of the results, and you have this file, which contains some basic information about the exam. So who was the patient, how much he weighted. Very important to check that the weight is correct, because otherwise the absolute quantification will be wrong. And you have also information about the time the PET took place and the time the injection took place. As I said, the injection takes place at a certain time, but the PET should take place 30-60 minutes after. We also can check the dose, that there has been no issue with the dose. Sometimes it can happen that there are, so we should check that we are in the correct range. Then the results. So for the SUV results, so absolute metabolism, we go into the SUV folder, and you have here a JPEG, a PNG actually image that contains automatically generated render of the SUV in this patient. Here it's not so bad, it's not a huge decrease. Then you have this SUV info file that contains the quantification of, at a global level, in this whole brain mask of metabolism in the patient on the left, and in controls here in column D. And automatically we have the computation of the overall decrease in our patient compared to control. So here we have a decrease of 30%. Again, it's not so bad. We can think probably we can lean towards maybe minimally conscious state. So this is for the SUV. Then for the SPM results, so the relative hypometabolism. So as I said, we need to do the quality check. And here you have the files for that. So here is the mask file I told you about. So you can open it with whatever you use for visualization. I suggest, for example, MRI-Prone, which is a free tool for visualization of nifty file. And you see here, we have a huge hole at the level of the left temporal lobe. So we know when we look at hypometabolism, these will be underestimated, will not appear in the results even if it is there. Then we can look at the spatial normalization. The last two files are the smoothed normalized images. and normalized images is the last one. And you can just take a look and see if you can also open the other template for comparison and see if there is any issues. Here, there is no issues. Everything is where it is, but we can see a huge lesions here on the left, consistent also with what we saw on the mask. Then we have here all the renders. That's the same that you saw also in the presentation. The last one is a render with the axial visualization that is useful again to look at some effects that we see on the 3D brain renders actually are located in the white matter, as is the case for part of the effects here. Then you have a series of Excel files, as I mentioned, that indicate, quantify the proportion of hypometabolic and relatively preserved voxels for a series of regions, networks, lobes, and also for regions of the language network specifically. So, okay, sorry, this is a video, so I'm trying to synchronize it. It's not super easy. So for the network results, we can open them, and you can see here, for example, we have here the amount of hypometabolic preserved voxels. For example, for the default mode network, we can say that there is 30% of the voxels that belong to the default mode network that are hypometabolic in this patient. These are just files that are meant to help with the interpretation of the renders. Is just, again, to provide a more accurate interpretation. These two files are files that contain the statistical maps that are used also for the renders. You can open them, overlay them on a template of your choice, for example, using MRI Cron or any other software, and you can explore a little bit more your results. Just, if you have any doubt, if the renders are not, maybe clear enough on some regions, you can quantify a bit better the effects, localize them precisely with this kind of help. And that's it for the interpretation. Just one thing, as you can, so it's a bit long to do all of these things, take the file, one file there, one file there, one file there. There is a premium feature which is not available, but you can ask us if you want to have it, where all these different files and renders and percentage of decreased SUV are taken automatically from the different files by the pipeline and are just used to create an automated report. So you just give a template of the report where we describe the analysis, how they work, and the pipeline takes the 30% from the Excel file, puts it here. Then we have something that is, of course, the description of the results needs a user, but this premium feature also allows you to take whatever renders you want for your report and just organize them into an already, well, almost, let's say, final output file. And I think now that's really it. So we did this for the PET report of the Coma Science Group, but that's it. For the last part, so I think, so the technical part, I think it's taken care of by the pipeline quite nicely. But again, as you can see, it's more tricky to provide a correct interpretation of the results. So if you want to know more, we had organized a workshop a couple of years ago, and all the recordings are here at this link. And, everything is available to if you want to go deeper into some aspects, the clinical reports, the quality control, or also the analytical steps. One last thing, very, very important. So if you want to use this pipeline, I think it's good to stress some good practices for adopting this pipeline in your center. Now, as I first mentioned, most steps in this pipeline are fully center independent. So this will work in any center. But both the control images that are used for statistical comparison and for estimating the decrease in SUV and the template that is used for normalization are things that have been acquired by the Common Science Group. So it is important to assess whether the data of your patient are comparable with the data, with these data that are provided with the pipeline. And the comparability must go over two different levels. First of all, the reconstruction procedures, so that the physical characteristics, let's say, of your PET image should be similar between the control groups included in the pipeline and your patient. This, I think, our physicist is always happy in general, Claire Bernard, to reply to questions about comparability in terms of reconstruction. In general, it is good to not use PSF reconstruction algorithms if you want to compare your patient with our controls. And it is a bit more tricky to use images derived from a PET MRI scanner, as our images were derived from a PET CT scanner. But it's not impossible, all depends on the reconstruction procedures that are used. Second, super important point is that even the acquisition procedures, so let's say the state of the patient, is kept consistent with the state of our controls after injection. So as I said, we don't start to scan right away after the injection. We have to wait at least 30 minutes. In these 30 minutes, it is super important that the patient keeps eyes open. Our controls were acquired with eyes open. The patient should have eyes open, otherwise you will get some hypometabolism at the level of the occipital lobe, which is purely artifactual. It's not due to the condition of the patient, it's just the way the scan was done. Likewise, the patient should be kept awake. So this is also why he should have eyes open. If he has eyes closed, we don't know if the patient is awake or not. And I've shown that under sleep conditions, we have a physiological decrease in glucose metabolism up to 20-25%. So that could change our evaluation quite a bit. Finally, super important, we need to be quite strict with the level of glycemia in the blood of the patients at the time of the injection. Different states have different guidelines that can be more or less permissive with the level of glycemia that is allowed during an FDG-PET scan. What we suggest is to keep the level between 80 and 100 milligram per deciliter. Because what happens is that if you do an FDG-PET scan, even in a perfectly normal, healthy control, you get this kind of pattern of hypometabolism. So an healthy control with a high level of glucose in their blood that will compete with the FDG tracer that we will inject will have a relative map of decreases that really looks like a map of an unresponsive patient. So if the glycemia is high, we don't know if our patient is really, has this kind of massive decreases in metabolism because of the glycemia or because of his clinical condition. So this is very important. So what happens if you discover, and this is more on the technical side of things, that the patient and the control data are not comparable? Well, the best solution, what I will recommend to any center that can do this, will be to acquire your own control data and create your own template. And it is very easy to add to adapt the pipeline to take in as input your own data. If you don't have a chance, because I know in many states, ethical committees are not too happy to allow control subjects to go under an FDG-PET scan, what you can do is you can look into the existing data sets. If you go in this paper, we did a huge work trying to categorize all existing databases that are PET databases that are easily to access. And we found more than 30,000 PET scans that you can access more or less easily. There is a chance that there is already some healthy controls data available that are comparable to the characteristics of the data that you are generating with your scan. Last but not least, we are acquiring at the Coma Science Group a new database of health controls, and we plan to keep the raw images so that it will be possible, under request, to perform some tailored reconstruction procedures to adapt our controls with your patients and keep, again, make the two comparable. So yeah, that's it. So I would like to thank everyone at the Coma Science Group, and in particular the people that were involved in these projects, Andreas, Nikita, Claire, and Jitka in particular. And I will leave you with the last slides, just if you want to know more. We also plan to organize a new workshop. At some point, you can already pre-register if you'd like to just keep up to date. So I guess that's it. Thanks very much for the attention.
Thank you so much, Arianna. This was a very helpful tool, and I think it needs to be even worked on more to being developed and being used by any center. But I think it's a wonderful first step. So thank you so much for your talk. It was extremely interesting. I have a few questions for you. So the first one is, do you have any plan for incorporating the calculation of the metabolic index in the pipeline too?
Yeah, so it was in the plans. Let's say that, I mean, there is a lot of things to do that I would like to do, but there is not many people working on PET in disorders of consciousness.
I know the problem.
So I want to, yeah, that's in the plans. It will be a bit more complicated because there is some more basic requirements that needs to be fulfilled. It's just not just MATLAB and SPM. There is more software that needs to be used. So yeah, it's in the plans, but I cannot give you any timeline for that for now.
Could you just very briefly explain, maybe to the registrants, what is the metabolic index?
Yeah, so the metabolic index, let's say it's an alternative way.
Shortly.
Yeah, I will not go technical. It's an alternative way to quantify absolute glucose metabolism. Why it has been created by Stender, the Coma Science Group? Well, it's because SUV had some limitations. So let's say in some cases, SUV might be not particularly precise if the acquisition procedures are not really kept very well under check. So with the metabolic index, we have an alternative way to quantify absolute metabolism that use extra cerebral signal to do the quantification, and just it's meant to be more precise than SUV. So it's an alternative, more precise way to do absolute quantification, but more challenging to implement. Hope it was short enough.
It was also an interesting discussion about how do you account for the difference of skull deformation and brain deformation in patients with severe brain injury.
Yeah, so as I mentioned, like not all the time the spatial normalization works. So in some cases, there is no way the spatial normalization will work in case really of huge deformations in the brain. It might work, as I showed, for some regions, and it might fail for others. Typically, the caudate sometimes doesn't work very well. I try to, I mean, there's a lot of work to do also in that respect. One could try different strategies. Like, for example, you can change a bit the parameters for spatial normalization. One could not use a template, but use the info in the structural MRI of the patient if you have it to do the normalization. But sometimes, I mean, in my experience, it's very hard, in very extreme cases to get a good spatial normalization. So it's an open issue, I would say.
Would you say, how many cases would you say that you encounter this type of difficulty usually, and where it's really not possible to use the pipeline because of that?
So among patients that then were diagnosed as minimally conscious, not that many, like I would say almost none. In patients that were unresponsive, I think it's very rare that I really cannot interpret any of the results. Most, I mean, I would say maybe in 1/5 to 1/3 of cases, there is some parts of the brain where the results are not interpretable. Again, thalamus, caudate.
Yes, patients.
Yeah. I cannot give, I don't have a feeling. What I want to actually, it's something I want to look into.
No, just a estimation, yeah, subjective estimation, pretty much.
It's not a majority, but it's a consistent number, let's say.
Exactly. Okay. So I have another comment that has actually 3 questions. Thank you for presenting. This is very interesting, especially since you are able to analyze smaller portion of the brain. A few questions. The first one, is there an exercise or mental task during the scan, or just resting? I suppose it's just resting, but I'm just asking in case.
So, I mean, with FDG-PET, you can do what you want, but indeed, all the results, all the literature is about resting state, which is also something like, it's kind of considered as it's easier to do an FDG-PET scan at rest than something that requires a more active paradigm. So it's all resting, yes. You can do also tasks, but it's not done, at least in the patients. I'm not aware of this.
And I know the answer, but I will ask anyway. Does it matter when the patient has eaten, whether they have had coffee or other stimulants such as Adderall, Memantine? amantadine, I suppose, and so on. I don't know what is memantine, but I'm not a doctor, so maybe there is such a thing.
Yeah.
Memantine.
So I mean, these are also in general. Health controls should fast at least 4 hours before the exam. For the patients, we suggest to do fasting at least 6 hours before the exam, because again, as I said, the reliability of the exam depends on the level of glucose metabolism in the blood. So that's really something you need to, I mean, if your nuclear medicine for sure will ask you to do that, otherwise you can just throw away your exam. It's pointless to do this. What effect of stimulants, coffee, whatever, again, in theory, you're not, again, in this time window, you're supposed just to drink water, so right before the exam, and wherever possible, there should be no drugs with effects on the brain that should be taken. So one should try to plan the exam to avoid this kind of effects. Again, I know it's not always possible, but it's a method of evaluating, is it worth it to do it even under this condition? But I guess the point would be to be aware of what the patient has taken before the exam to account for that.
Yeah, anyway, I mean, there are procedures that, the radiology will ask you to, definitely having a patient that is not fed is something that is very important, and will be normally will be pointed out by the radiology department when you're doing a PET scan.
Yeah, absolutely. I mean, there is specific guidelines to follow for FDG-PET exam, so yes.
Exactly. And so finally, do you believe to be the most appropriate time to use the scan immediately after injury, later, only very severe, many months after injury, or other time points?
So
just a question about time points.
Yeah. So how we use it here, it's really in prolonged patients with disorders of consciousness. So also all the reference literature, it's available for patients in a prolonged DOC with very different time points. So I think, I don't know, I think it's important to have it, maybe not too late, because again, as it was shown by some studies, it can help understand predict better the prognosis of the patient. I've not had much experience with acute cases, actually. I know you can use it, for example, in a patient in coma, because again, just having the eyes closed, you don't know, again, if it is awake, if it is asleep, so it's a bit challenging. In acute patients, it can be hard, especially if they are in the ICU, because anyway, it's not something you can do at bedside. You have to take the patient to the scanner, and that, of course, constitutes a risk. So I guess there is evidence really for utility in prolonged patients with disorders of consciousness.
Thank you, Hannah. And my last question, which comes from me, is what's the next step for your pipeline? Do you have any thought about implementation, things that would help to understand how much barrier there is for using the software?
Yeah, I mean, there's some
that would help clinicians
to do. Yeah, so there is more functionalities I'd like to add if I ever had time. But let's say for now, yes, for sure it will be. So now the pipeline is being used right now in 2 other centers, one in Germany, one in China. So and they're using it independently from us. But for us, yeah, for sure it will be interesting if anyone has access to PETs of QC patients to try to implement it again in a clinical context. For now, I mean, in my case, it was not so. I mean, it was not too difficult implementing this in other centers, but these are really our collaborators. So it will be interesting to see in the real world how the pipeline goes. So if you want to try and let me know, I will be very happy to.
And you know that I'm interested, so I will definitely be in contact with you. I would love to have our doctors actually just use it with some guidelines, of course, and see how that goes, and come back to you with different commands and things to
many things
help you
many things I would like to do are also similar to the questions. I would like to try different types of normalization, so allow different strategies. If it fails, what can you do? But yeah, it will take some time and some research to see what works best and then implement it. So
wonderful.
Thank you very much.
Well, Arianna, Dr. Sala, thank you very, very much for presenting today. This webinar, it was recorded and is recorded, and will be available to all the registrants, also other members if they want, I think, to have access to it retrospectively. I think that might be possible, but I don't want to say something that is not. Jennifer, is it correct? Jennifer?
Probably.
Hi, I'm sorry.
Okay.
Are you ready
So-
to depart, or was there a question?
No, I was wondering if members want to access this retrospectively, even though they were not a registrant, if they can.
Yes, eventually we will have it online for everyone to access. But yes, if members want to get in touch with me, I can get that to them. So it's jwiley@internationalbrain.org.
Great. Thank you very much for hosting me.
Thank
Guys,
you.
thank you so much, everyone. And I think we'll have another one in January that will be most likely on family education booklets that was developed by the ACRM. And so I hope that I will see you at that time. Have a great day, everyone, or night, anywhere you are.
Thank you.
Yes. Bye-bye.
Bye, everyone. Bye.